We started our project by looking at a paper by Fernandez-Rodriguez et al., published in 2017, Engineering RGB Color Vision into Escherichia coli. And as the name implies, the lab incorporated color vision into E. coli, and the color response system was also put into the same strain. So the bacteria could sense color and in response make the same color. The system put into the E. coli is very complex, comprised of 18 genes, 14 promoters, 18 terminators, and 4 plasmids, the total being 46,198 base pairs of DNA. The system is comprised of 4 parts: the sensor array, which receives the light and recognizes different wavelengths, the circuit which processes the light signals, the resource allocator, which connects the circuit to the actuators, and the actuators, which produces the colored pigment. These are the actual images of their bacteria culture, producing the image shown onto the culture. The paper also showed other potential uses of this system. For example, by changing the actuators, the last part of the system, the strain could produce desired effect upon receiving different light signals.
Represented here is the overall schematic of the red, green, and blue system, as described by the original authors of the paper. The red sensor array system is comprised of a histidine kinase and a response regulator, where the histidine kinase is the CP8H* protein. This protein is a chimera protein that is comprised of a red light sensor domain and a histidine kinase domain, as taken from the protein ENVZ. The histidine kinase domain will phosphorylate the response regulator, which is OMPR. OMPR, in turn, will promote the transcription of the protein sigma KIF after follow-through of a NOT logic gate. Since the green sensor system would be rendered useless with the addition of the yellow sensor system, rather than removing the components of the green system, it was decided that the green sensor system would be modified and utilized to regulate the red sensor system by controlling production of the CPH8* protein. The proposed methodology involves utilizing a site-specific recombination system that is naturally found in uropathogenic E. coli, which from here on out will be shortened to UPEC. Certain UPEC E. coli have a Fin operon that is comprised of Fin proteins that contribute to either the construct of a pili or a fimbriae. This operon is regulated by 2 site-specific recombinases, FimB and FimE. These proteins target inverted repeats, as noted in the diagram as IRL and IRR. By acting on these inverted repeats, they can flip the promoter into either the on or off position. In order to utilize this system, 2 major changes to the red and green sensor array would need to be implemented. First, the CGG sigma protein gene will be replaced with the genes for FimB and FimE, while retaining the promoter already present in the plasmid. Second, in order for these proteins to be able to act on the promoter of the CPH8* protein, inverted repeats will also need to be added upstream and downstream of the promoter. Implementation of these changes would allow repression of the red sensor system only when green light is present. Shown here is theoretically how the CGG gene would be switched out for the FimB and FimE genes. First, UPEC chromosomal DNA would be extracted and purified by a phenylchloroform and ethanol precipitation process. Next, traditional PCR and an overlap fusion PCR reaction would be implemented to amplify FimB and FimE, and then combine these 2 gene products together. Once this is done, a 2-step homoendonuclease reaction would then be conducted in order to switch out the CGG gene in exchange for the FimE and FimB construct. The main difference between the homoendonuclease reaction being executed in this step and what we have learned about in Nano's lectures is that the 2-step homoendonuclease reaction is occurring within the plasmid, not within chromosomal DNA. Hence, the ISCE1 site alone would not be sufficient for selection for the cells that contain the construct of interest. Either a second restriction site would have to be added, which in turn can be utilized for a fragment restriction analysis of the plasmid, or alternatively, replica plating can also be implemented to determine the cells that are susceptible to kanamycin. The next slide shows the overall scheme for how the inverted repeats will be added to the CPH8* promoter. The main idea here is that rather than adding inverted genes directly to the promoter, it was decided upon to approach this in a rather convoluted manner. The best way to think about this is as the following. Rather than adding inverted repeats to the promoter, the CPH8* promoter will be added to the inverted repeats that are already present within UPEG chromosomal DNA, and in that segment that contains the CPH8* promoter and the inverted repeats would then be amplified and inserted into the original location within the plasmid and where the original promoter was. This would be done very similar to what was done in the previous step, involving a lot of PCR reactions and several homoendonuclease processes. The main reason this methodology was decided upon was because, as indicated in the slide here, there are other elements within the promoter that are utilized in vivo to help increase the efficiency of the promoter flipping. Rather than conducting a ridiculous amount of PCR reactions to add these additional elements, or to not include these elements altogether, substituting the important parts of the FIMS promoter, i.e. the -35 and -10 portions, with the CPH8 counterpart, would theoretically allow the inverted repeats to be present, but also allow some additional elements to be present in vitro that would help the efficiency of the promoter flipping when the red repression system is activated.
When constructing the yellow photoreceptor pathway, it became apparent that there are no naturally occurring photoreceptors that are specific for yellow wavelengths of light. As a result, we took a more broad-spectrum photoreceptor and modified it until it was activated exclusively by yellow wavelengths of light, around 550 nanometers. We used the photoreceptor animal-like cryptochrome, which is found in the green algae Clitomonas reinhardti. Cryptochromes are a class of light-sensing protein that regulate gene expression and are mostly found in plants and other photosynthetic eukaryotes. Cryptochromes, like most photoreceptors, are either bound to the outer membrane or found in the cytosol. Activation leads to a phosphorylation cascade that can lead to the repression or promotion of multiple gene targets. Animal-like cryptochrome is responsible for the promotion of several genes simultaneously. One of these genes, GLN1, encodes glutamine synthetase, which has a specific repressor gene which will become important later. We added DNA encoding a yellow protein to the end of the GL1 gene to make it produce a yellow color when activated. When choosing which yellow protein to use in this insertion, the exact color of the protein was very important. The color range of our experiment depends on the mixing of the three primary colors, and as a result a standard yellow color needed to be chosen. Yellow fluorescent protein, though by name sounds like the obvious choice, is actually slightly green in color and would affect the colors that yellow is involved in producing. M-banana shows the best coloration and was inserted into the GLN1 gene. Once the code for M-banana had been added to the GLN1 gene, the cryptochrome then needed to be mutagenized so that it was only activated by yellow wavelengths of light. Alanine scanning mutagenesis was performed on the animal-like cryptochromes to find a mutation that would allow for a narrower range of activation to yellow wavelengths between 540 and 570 nanometers. Animal-like cryptochrome is normally activated by light in the red, blue, and yellow ranges. To screen for a desirable mutant, cells were isolated, grown, and replica plated. Each plate was then treated with a different wavelength of light. As the M-banana protein is produced when animal-like cryptochrome is activated, cells turned yellow in response to this activation. This allowed for an easy visual determination of successful mutations. Colonies that turned yellow only in the presence of yellow light were selected from these colonies. The now modified animal-like cryptochrome and the GLN1 gene with the M-banana attached were then isolated using gel purification and combined using fusion PCR. Overlaps on each of these genes then allowed for an easy insertion into a plasmid for the transformation into E. coli. Plasmids were inserted into E. coli using electroporation. Screening was then done for successful transformations. As successful transformations were characterized by yellow protein production in the presence of yellow light, isolated colonies of successful transformations were easily identified. Now that an M-banana protein production pathway has been set up, a repression system also needs to be established. Yellow pigments are required in the production of all colors that lie between red and yellow and blue, but would disrupt all the color formation that lies between red and blue. To make sure that yellow pigment isn't produced when these colors are to be expressed, a repression pathway was constructed. This pathway was activated by violet light, as the color violet requires no yellow pigment to be produced. The violet photoreceptor that we chose to use in our pathway was first discovered by Ramkishan and Tabor, and uses the UIRS and the UIRR subunits to increase the transcription of CSIR and ISIR. URS is a membrane-bound histidine kinase photoreceptor that, when activated, phosphorylates URR, which in turn promotes the transcription of CSIR and ISIR. Ramakrishnan and Tabor showed that by cleaving off the CSIR1 and ISIR genes and replacing them with superfolded GFP, it is possible to create a gene expression that is regulated by violet light. Their study found that increasing light intensity also increased gene expression, with the opposite effect shown with decreasing light intensity. This is an ideal system for us because it allows us to revert back to a yellow production pathway if we change light sources. In our project, we replaced their superfolded green fluorescent protein gene with our GLN1 repressor gene. Ramakishan and Tabor also wanted to show that they could easily transfer their gene expression system to another organism. To do this, they added their genes to a PPR plasmid using Golgi gate assembly. They used a two-plasmid system as they found that it gave a higher percentage of successful transformations. As we would be using the exact same system, transferring our plasmids to E. coli, we decided to copy their plasmids, exchanging our GLN1 repressor gene for their superfolded green fluorescent protein gene during the Golgi gate assembly. Following plasmid construction, yellow protein producing E. coli cells were once again electroporated to allow for transformations. Isolated colonies were then exposed to yellow light to ensure that the yellow protein production pathway had not been disrupted. These yellow colonies were then exposed to violet light. Colonies that lost their yellow color were determined to have successfully taken up all three plasmids, completing the yellow production and repression system.
So now we are going to do the blue construct. Proposal of the blue construct. We already have the blue pathway that produces blue pigment in the presence of blue light. In addition, we need to arrest or appreciably reduce the production of blue pigment in the absence of blue light, characterized by the presence of orange light, so a wavelength of 590 to 620 nanometers. The blue light sensor signal array is connected to a NOT gate. This is a type of logic gate used in engineering and now applied to methods in synthetic biology in which an input results in no output. In this case, the input, the blue light, drives the expression of a repressor which turns off the output promoter. Knowing that turning off the gene expression is what produces the blue pigment, we looked for a mechanism that would ensure that the gene expression is kept on in the absence of blue light and also in the presence of white light. There is no two-input logic gate that covers this scenario. Instead, we will need two consecutive NOT gates with the upstream system sensor for orange light. Phytochromes are currently being characterized in algae that span the visible spectrum. The prasinophyte phytochrome NPYR PHY1 from Nephroselma pyriformis specifically senses orange light at 584 to 614 nanometers. This phytochrome was sequenced and its PIF signaling pathway was coupled with its gene activation promoter and GPBE box. This was placed upstream of a flavin mononucleotide FMN riboswitch that is bound directly to the FMN region of the blue sensing YF1. Activation of the riboswitch creates a loop in the FMN transcript, halting translation of the blue light sensing component and removing the ability of YF1 to sense blue light. This causes the gene expression to remain active, producing no blue pigment. Thus, only in the absence of orange light can blue pigment be produced. UV fixing using epoxy resin. We used epoxy resin to fix the bacteria after they reach their maximal color profile. Epoxy resin contains two parts, DGEBA and IPD. In determining what kind of resin to use in the fixing of cells, it was important to consider that the cells must remain intact while also preventing DNA damage, membrane damage, and damage to the synthetic systems that allow for the color production. In their 2009 paper, Aoki and Ichimura showed that including oligomers into the epoxy resin allowed for more rapid fixing while also limiting UV exposure of the sample. We tested 4 different oligos to determine which gave the fastest fixing time, as well as the least amount of damage. The most efficient oligo was experimentally determined to be the tetramer shown above, with the most rapid fixing time and the least amount of damage done to the cells. Prior to fixing the cells, it is important to ensure that the colonies have produced the maximal amount of color in order to ensure significant pigmentation of the final fixed product. After monitoring our various colored colonies every 5 hours over the course of 24 hours, we determined that the maximal color production occurs at 20 hours. We used this data to time the fixing of our final product shown above.
The cells that we have created in this experiment showcase the potential that the field of optogenetics has to offer. Many new in vivo experiments involving cell morphology, gene expression, and cell signaling can now be controlled purely by light sources. This has created a simple, cheap method to generate specific responses in cells. This methodology has even been transferred to mammalian cells, which has given rise to potential biomedical applications, as light can be a non-invasive trigger to target very specific cells. To give just one example of these potential applications, researchers have already managed to create an artificial pathway in type 2 diabetic mice that uses blue light to increase the glucagon-like peptide 1 expression, leading to insulin secretion and returning the mouse to glucose homeostasis.